Hi everyone, welcome to the Biologically Important Molecules or Biochemistry Lab. First of all, can you read the instructions? I know they're a little long, but I strongly encourage you to read them all. And at the end there's one question for you to answer. Pause the video and answer the question. The first test we're going to do is the iodine test. So I have iodine, water, starch, and I've created slurries of onions and potatoes. Remember, water is your negative control and starch is your positive control. So remember, in your chart, you're going to record the color the iodine changes, and from those results, you're going to tell me if there's a presence or absence of starch. So first the water. Then the starch. Quite a bit different color there. Then I'm going with the onions. And then I'm going with the potatoes. The potatoes are slightly darker than the onions. You'll notice that they're getting darker and darker over time. For the next experiment, let's put on some safety goggles because we're using boiling water. Okay, so now we're doing the Benedict's test for reducing sugars. Just like before, you're going to record the color of the Benedict's and whether there's a presence or absence of reducing sugars. Now, the catch with this one is you have to wait until it's boiled to record the colors. Wait until it's boiled. So, we have water, negative control. We have glucose, positive control. We again have our onion and then our potato. So we'll add the Benedict's. Remember, you're not recording the color yet. Put it in the boiling water. Good idea to label what is what. Okay, so it's been a good 5 minutes and I'm going to try to carefully remove. So with water, this is the color. With glucose, this is the color. Onion, this is the color. And let's just have a point of contrast. So this is blue with the water and it's greener with the onion. And then lastly, we have our potato. And that's a real pale green compared to the water. Okay, so now you have done all the carbohydrate tests and you need to pause and answer the questions. Next, a test of proteins. We're using Burette's solution this time around. Water is our negative control, egg is our positive control. Then we have our unknowns, gelatin and flour and water. I think I'm going to have to use a lot. We're going for the big bottle now. Okay, so hopefully you noticed a bluer, light blue color for the water. I think you can see that color by the egg there. Gelatin. And finally, our flour. Make sure you record the colors and if they contain protein. Please do your questions on proteins. Last test is for the lipids or the fats. So this is probably the least scientific, but we're going to go for it. So we've got brown paper towel and we're going to smear substances on them and then record if they're translucent or not. So translucent, basically we're looking for greasy and shiny. So first substance is water. Next substance is oil. So negative control, positive control. Then we're doing whipping cream, and this would be milk product A, whipping cream. Smear, smear, smear. And then I have Philadelphia cream cheese. Smear and smear that around. Okay, let's see when we hold it up to the light. Woo! Boy, is that oil greasy and shiny. Can you see it okay, Filmer? Yes. Next, for our milk products. I would say that the whipping cream is a little bit more translucent and shinier than the cream cheese, although the cream cheese has a little bit of translucence. Record your data, finish your lipid questions, and you are done the lab.